there's an interesting flow of membrane from one vesicle to the other. So, for example, as the endoplasmic reticulum sends a transport vesicle to the Golgi apparatus, it's actually sending a membrane because membrane surrounds that vesicle. It fuses with the Golgi apparatus, adding membrane to the Golgi apparatus. So this membrane flow is a continuous movement of membrane from one organelle to another. So the membrane is easily replaced. The membrane, the outer membrane of the cell, that main plasma membrane grows and gets smaller as vesicles enter and leave the cell. The mitochondria is the powerhouse of the cell. It is responsible for cellular respiration or aerobic respiration. It's aerobic because it requires oxygen. It is in the mitochondria that glucose will be completely broken down to carbon dioxide and water, forming ATP. Cells have hundreds to thousands of mitochondria. Some cells have no mitochondria. Here's the mitochondria with its double membrane system. The outer membrane surrounds the organelle. The inner membrane has inner folds called cristae, and the enzymes for the reactions of cellular respiration are on the cristae. The liquid inside is called matrix. This is a brief visual of what's happening with cellular respiration. The first step of cellular respiration, and we always start with glucose, although other molecules can enter the mitochondria to be broken down completely, but glucose is the easiest. We start with glucose, and in the cytoplasm of the cell, glucose will basically be energized and split in half to 2-pyruvate. This first step is called glycolysis. It produces 2-ATP, but it is anaerobic. It does not require oxygen. The pyruvate will then enter the mitochondria, entering the citric acid cycle for a complete breakdown. Every carbon of the glucose, and glucose has 6 carbons, every carbon will be expelled as CO2. That CO2 is going to enter the blood and be exhaled from our lungs. Oxygen is required. And for one molecule of glucose, basically 36 to 38 ATP are formed. Again, that first step of cellular respiration is glycolysis. It occurs outside the mitochondria where glucose is basically split in half and pyruvate enters the mitochondria for complete breakdown in the citric acid cycle, complete breakdown to CO2 and water. The ATP produced in the mitochondria can migrate to anywhere in the cell to be used. Once it's used, the ATP has lost a phosphate group and it becomes ADP, adenosine diphosphate. That ADP has to go back into the mitochondria, and as glucose is broken down, the energy is trapped in that high energy bond, forming ATP again. So many pounds, it's estimated that many pounds of ATP are formed in our bodies per day. It's the only energy molecule that our cells use. The nucleus is where the DNA sits. The DNA is stuck there, just like a reference book in the library. You can make copies. The librarian won't let you make a complete copy of the DNA of the library book, but maybe photocopy a few pages. And that's what happens. The DNA is copied, small pieces of it are copied, and that's called RNA. That RNA will leave the nucleus because the nucleus is surrounded by a membrane with pores, and thousands of different kinds of proteins are produced because the information in the DNA is specific to making proteins. Most cells have one nucleus. Skeletal muscle cells are multinucleated. Red blood cells do not have any nucleus. So the cell responds to its external environment in preserving homeostasis. So things can happen in the external world of the cell, that is in the extracellular fluid, and those changes, there may be a hormone present which might bind to a receptor protein, enter the cell for short-term adjustments. There may be diffusion through membrane channels into the cell, direct interaction with the cell DNA to make certain proteins or stop the production of certain proteins to maintain homeostasis. The nuclear envelope is a double membrane system with pores to allow substances to pass out or into the nucleus. In the center of the nucleus, or somewhere in the nucleus, will be a round structure. It will look darker, and that is called the Nucleolus. That is where ribosomal RNA combines with proteins to form ribosomes. The ribosomes leave through the nuclear pore and will make proteins out in the cell. DNA is the instructions for making the protein. And DNA, for most of the life of the cell, is in a form called chromatin. Chromatin is the stretched out form of DNA, kind of like cooked spaghetti. Here. Well, this is really inconvenient for a cell when it's all stretched out. It's a huge molecule. Every cell in our body has about 6 feet of DNA in it. And it's real hard for a cell to divide and split up all that DNA easily when it's in chromatin form. So what happens to the chromatin is it starts to wind around these histone proteins. It winds around 8 histone proteins, forming a nucleosome, and continues winding and coiling to form the denser, thicker structure we know of as a chromosome. So we see chromosomes in dividing cells and loosely coiled chromatin in non-dividing cells. Every cell in our body has 23 pairs of chromosomes, 1 pair from mom, 1 pair from dad, and the instructions in the DNA are for making proteins. This is showing that this is what the chromosome looks like. This is a replicated chromosome, so it has this chromosome has made an identical copy of itself. It's held together at the centromere. Protein synthesis is called translation. It's a pretty complex operation. If we start with the DNA, if you'll recall, DNA consists of chains of nucleotides. If we use the analogy of looking like a ladder, the sides of the ladder consist of alternating sugar and phosphate groups, and the rungs of the ladder will consist of the nitrogenous bases. On the rungs of the ladder, the bases are paired complementarily. So adenine is always paired with thymine, cytosine is always paired with guanine. So here we have, so let's just say we've got a piece of DNA with AAA. Its complementary strand would be TTT, and this is the coding strand. That means this is the strand we're going to use to make proteins. So a copy is made, the strands open up, a copy is made of RNA from the DNA coding strand. Now remember, from the DNA template strand, there is no thymine in messenger RNA, but uracil replaced thymine. So the template strand is what is made a copy of. So UUU, it would be the codon for the messenger RNA. Transfer RNA requires an anticodon, and transfer RNA carries the amino acid phenylalanine. This is the amino acid. There's 20 amino acids in the body, and each amino acid is determined by the DNA template strand. So DNA consists of genes, which is the functional unit of heredity. For example, there is a gene to make insulin, and it is all the different regions. It's a specific region of the DNA that has the information to make a complete molecule of insulin. in order to make the protein, you have to uncoil the DNA. It must be in chromatin form. You have to uncoil the DNA and remove those histone proteins that it's wrapped around. And 3 different types of RNA are required. First, the messenger RNA. The messenger RNA is the copy of the DNA as RNA. The RNA is made into one strand. This is called transcription. And the bases would be adenine would pair with uracil, guanine with cytosine. The information is in triplets or codons. That means that 3 nucleotides in a row is the information for one amino acid. Now the amino acids have to be brought over to the growing polypeptide strand, and that happens using transfer RNA. So let's look at this visually. It's a lot easier visually. So here's our DNA strand. It's going to open up, a short section will be opened up here. Certain enzymes will do that. Then you're going to make, one copy is going to be made of one strand of the DNA, and that's the RNA. This is messenger RNA with the information to make a single protein. The messenger RNA leaves the nucleus and goes out and attaches to the ribosome. Okay? So all the enzymes to make proteins are inside that ribosome. So here is our messenger RNA. We'll pretend it's in the ribosome. You can't see it if it's in the ribosome. So here are the codons of the messenger RNA. The way the correct amino acid comes over is transfer RNA has an anticodon. Here the anticodon is UAC that will bind to the codon of the messenger RNA. AUG is complementary binding, and it will bring over the correct amino acid. So each transfer RNA brings over an amino acid. The peptide bond forms between each amino acid, and you have the formation of a polypeptide. Here's a summary. The DNA triplets determine the sequence of messenger RNA codons. The messenger RNA codons determine the sequence of transfer RNAs. And the sequence of transfer RNAs determines the sequence of amino acids in the polypeptide protein.